Welcome, Charlotte, today. I'm Frank Cavanaugh. In our continuing pursuit of your good health, we're going to talk today about the promise of ending degenerative and debilitating diseases. Today, millions of Americans suffer from the effects of stroke, heart disease, Parkinson's, Alzheimer's, spinal cord injuries, diabetes, kidney disease. The list is indeed a long one. But there is also today the promise of a revolution, and we may be on the threshold of being able to cure these previously incurable diseases. On one hand, it may be as simple as letting the body regenerate organs that are damaged, or amazingly enough, even the ability to grow replacement organs. Our guest for this program is Dr. Paul Sandberg, and he is a neuroscientist who is professor of neurology and director of research in the Department of Neurosurgery at the University of South Florida, and he is also a past president of the American Society of Neurotransplantation. Dr. Sandberg's early work was in the research on Parkinson's disease, but when his father suffered a stroke, his priorities changed, and much of what he learned may help to treat some of these debilitating diseases, and we may in fact be on the threshold of a revolution in medical care. Dr. Sandberg, nice to have you with us. I'm very pleased you're here.
Thanks. I'm pleased to be here.
Your research on Parkinson's disease, did you learn something about that that then had an application when your dad had his stroke?
It's a good question, because I think what we've been working on is the role of replacing dead brain cells that occur in Parkinson's disease with new brain cells. And the question is, where do you get these brain cells? And people have looked at fetal tissue as one approach, or a number of other sources, animal cells. And the work had been progressing quite well, especially at the University of South Florida, where we've been in clinical trials trying to replace these dead brain cells in Parkinson's patients with new cells. And I guess the question really related to how that affected the issue of my father.
Yeah. It was sort of an easy jump then, wasn't it? I mean, it was very logical to say, why can't I do this?
You'd think so. I mean, the jump then really was, stroke is a lack of blood supply in the brain. It kills off so many of these brain cells. And the question is, if they're gone, can we put new ones in? And when I was, a stroke happened out in Texas and in Houston at quite a good medical center. And I was there for a number of weeks with him and just saying, what about all these new therapies? Why don't we try something new, you know? And it was very conservative, very, methodical.
Right.
But none of this stuff had really made it to the clinic. And stroke affects so many people. So when we finally went through the episode of my father got a little more stable, I went back to work. We thought in my laboratory, why don't we start working on stroke? Why don't we look at the ability to put new cells in stroke patients?
Yeah. I had read, I guess my first reading or knowledge of stem cells was that they were used with cancer patients as replacements. Was that prior to your dad's stroke, or did the use of stem cells in treating cancer patients follow that?
Well, I think the idea of cell, if we want to call it, quote, cell therapy or cell transplantation, cancer is one of the big ones because people have done bone marrow transplantations.
Right.
It's not unlike doing a bone marrow transplant in a child with leukemia or an adult. But in this case, I think at that time most people were looking at fetal tissue. Can they take tissue from, fetuses that were not there, the ethical considerations being, of course, there, and can they take these cells and put them in? And unfortunately, because of logistics, ethics, morals, the whole issue, the question is, could they find other ways to find tissue? And then they went to animal cells. Can we use fetal pig cells? And there you have a lot of immunology problems, that is, rejection problems of the new tissue. So it got to the point of a stem cell. And that's where we've kind of focused ourselves today, on these stem cells. And that's really, I think, what you're trying to get at then.
Right. Help us understand, what is a stem cell?
Okay. A stem cell really is a cell that creates the real stem cells, like the cell that kind of creates the whole body. It should be really called in some sense a root cell, shouldn't it? In many ways. But it's really a stem cell. And so if you think of the sperm and the egg combining, and they make a number of cells, they divide and they make they get to a certain point where one cell can still have the capacity to make any organ in the body. And that is probably the true stem cell.
Okay.
Then, as these develop in an embryo, what happens is these cells become organs or become tissue, and then they become what we call progenitors. They're an early cell, a stem-like cell, but they can only create a few different things. And this is the area that we pretty well tackle in brain research, is these progenitors, these cells that can become any cell in the nervous system. It's an early cell.
I saw that program that you did in Great Britain, and I thought I was looking at a piece of science fiction when I saw some tissue that started to grow hair and teeth.
Right.
And you wonder if you're being put on, and you say, my Lord, this is the most amazing science that I have run upon.
It's an interesting tumor. It's actually a tumor that occurs called a teratoma, or if it's cancerous, a teratocarcinoma. And it's in many ways like a stem cell tumor. It's actually a tumor that derives from early cells in the testes, for example. But they can occur anywhere in the body, these cells, and when they develop and become a tumor, they grow any kind of tissue. They can be bone in there and hair and all sorts of things, and nerve cells. And what's ironic, I'm just thinking about it now, is those are the cells that we've been looking at first to try to create replacement cells for stroke. These are cells derived from those kinds of tumors, treated in a certain way, that only make brain cells. So as we treat those cells, everything else dies off except for the brain cells, and that's what we've been using. But what's interesting, I hadn't thought about it till now, is my father had a teratoma. There was one tumor he had that activated once in a while. They cut it out a few times, and it was a teratoma. It was non-cancerous, but he would almost like say, "This is my twin, this tumor," because it had bits of hair and things in it. So it was interesting.
Oh my. We'll be right back after these messages, and we'll talk to Dr. Sandberg about cloning and how this is similar, different, those kinds of things. Stay with us. Welcome back. We're talking with Dr. Paul Sandberg, a neuroscientist from the University of South Florida, about stem cells, or as we're trying to understand them now, about root cells. It was several years ago, I guess, that Dolly the sheep was cloned, and then not too long ago there were 5 little piglets. Is all of this related at all to cloning? Is there some similarity in what you're doing and to the cloning process?
Well, in essence, I think there can be. I mean, when you think of cloning, you're thinking of a whole animal, a whole being made from a single cell that's been fertilized. And I think what we're interested in, to that extent, may be called tissue cloning. We're trying to clone new tissue in this area. They call it tissue engineering, in fact, as a field. Can we create new types of tissues? Brain tissue is the one we focus on, but there are a number of other types of tissues you can imagine.
Yeah. Food and Drug Administration looks upon this kind of research how?
I think the Food and Drug Administration looks upon it favorably as a potential. They have a branch at the FDA that focuses totally on tissue engineering, cellular replacement therapies. On the market right now, there's really only a couple that have made it at this point, which is skin, where they've have skin transplants, tissue available. But there are a number of companies on the horizon that are, in fact, developing new types of approaches for diabetes, for liver diseases, for brain diseases, by creating new cells for replacement. And it wouldn't surprise me in 5 to 10 years that a number of these will be on the market.
Okay. Your work in the beginning was with animals. What kind of results did you get? What were you able to do?
Well, it's interesting. Our first study we did when we got back to the lab after that trip to Houston was related to making stroked animals. And how do you make a stroked animal? For those people that may or may not believe in animal research, in many ways, if you're going to look at a stroke, you really need to use an animal that will show a stroke. And we used rats in this case. And we created a stroke as if a stroke might occur in a person by occluding or blocking a certain artery that goes up into the brain. And we found that, in fact, that these brain cells died off, and there were, in fact, symptoms that these rats had. Now, the question then was, could we then put new cells into the damaged area and get recovery? And we used fetal tissue because we had been used to that, but we also started using these neurons that were derived from these tumor cells that I talked about earlier. And this was a whole unique issue because these were human neural cells, human brain cells grown in a petri dish, in the test tube basically, and implanted into these rats and seeing whether they would work. And in fact, what we found was that the fetal cells seemed to work, and that had been reported previously by some other groups, but these new human neural cells that were grown in a test tube, basically, put into the brain of these rats. Within a month, those rats started walking normally and showing recovery.
My goodness.
It was quite interesting.
My goodness. Now, you have developed a relationship with Latent Biosciences.
Mm-hmm.
What is that, and what are the two of you trying to do together?
Well, I think the interesting part about this was that we do run a meeting here in Florida, in Clearwater. I know it's not in Charlotte County,
Right.
but it's close. And we run a meeting where it's the American Society for Neural Transplantation and Repair. And a number of international scientists and U.S. scientists all come down once a year for this. A number of companies that are related to this come there. And I was sitting by the pool having a iced tea and was talking to a president of this company, Gary Snabel, who had these cells, these human neural cells, that he felt was very unique. They had a patent on these. And he thought they might be useful for transplantation. In the meantime, what they were using them for was for drug companies to look at toxic chemicals. You know, so here's some brain cells grown in a dish. We can test toxic chemicals on these without having to go to animals or other trials. So we thought, okay, why don't we try this in the stroked animals? I thought these could be the cells we could try. And so he sent us some cells, and that's when we got those results. And so then he developed a relationship with us at USF in developing a transplantation program that we could take those cells, hopefully, to patients to do that. Because, as you know, government money is hard to get. And in many ways, in order to push products to the market for people and patients, you need companies behind you that have some financial means.
Right. You then took those to human patients and designed a protocol or applied that research?
Well, the first thing, of course, is to go to the FDA.
Right.
Go to the FDA and say, hey, this is what we want to do. And what do we need to do to show safety, no toxicity, and to test the possibility this may work. And so they gave us a number of studies in which to do, mostly animal and test tube type studies to show safety. And then we designed a first clinical trial that was actually done at the University of Pittsburgh. Why wasn't it done down here, you might say? But the main reason is University of Pittsburgh really was one of the transplant meccas in the United States.
Okay.
And and they also have a big stroke center up there. And so we felt the combination of that, and Doug Consioka, the neurosurgeon up there, was the first one then to implant a patient with these cells. And the unique part of that was the first time a patient had been implanted with stroke, with new cells, and the first time human brain cells grown in a petri dish. So you wouldn't have any ethical considerations about fetal tissue or animal cells or any of this were put into a patient as well. So it was quite an interesting study.
My goodness. Attitudes of those patients and their families about this possibility is what?
Well, another good question, because when you have a sufferer with any kind of debilitating disease, and these are patients that have had, you know, the FDA is not going to say go in any patient. They're going to say, okay, but you have to go into patients that have shown no recovery from best medical care and that are very severe. So these patients are paresis. That means that they're paralyzed on one side of their body. They may not be able to talk. They're really very dysfunctional. And so those were the patients we went after, the toughest cases. And of course, people want to help their family members, and they want to help themselves. And so, you know, they're looking for anything in many ways. And so it wasn't hard to find patients. And in fact, once the study was done, the first 12 patients and the results were announced, or even the first patient was done and it made the national news, there must have been 1,000 odd emails and requests that came in from families around the United States that you know, this was hope for them, because in stroke, a third of the patients die when they get strokes. A third get better, and a third show permanent deficits that really affect their lives.
Mm-hmm. What were the results? What was the outcome of the first study?
Well, that's just being published in a couple months, actually.
Okay.
They presented some data recently at a meeting, the Pittsburgh group, and they found that, in fact, out of the 12 patients, and I'm just paraphrasing, and please, you know, really wait for the results to come out, but about half the patients showed some improvement. These were patients that had no improvement whatsoever for a number of years, or whatever the average time was, and they showed some improvement. Again, it's not a double-blind trial, so it could be anecdotal, or it's an open trial, in other words. Could be based on hope, but in this case, they really had not shown any benefit for a number of while. But the most interesting thing is, so you might say those symptoms, well, the critics will say, well, but maybe they were really just trying hard, okay? But when we looked at the brain scans, and we looked at what we call PET scans, that is, these are scans of the activity of the brain. And in the stroked area, it's like a black hole. There's no activity. But when you looked at those patients that showed some recovery, there was new activity in the brain of these patients, which really is hard to relate to, the cells not working in many ways. But what it suggests then is that there's promise, and that there needs to be a larger study to look at perhaps dosing, because it was really only very low dosing at that point. And then after that, do a double-blind, placebo-controlled trial like the FDA wants in order to approve something for the market. And that's really where it's at right now.
But with this first group of 12, in the condition that they were in, one would not have expected to see any change in half of them.
Exactly, exactly. These were the worst cases, and if anything, they might get worse.
The potential of all of this, if it works, goes to where for what kinds of other problems? I mean, I said at the outset that there was the promise here of the end of degenerative and debilitating disease. Is that too strong a statement, or do you feel that?
Well, you know, if we look through the century, the next century, maybe it's not too strong of a statement. Things always take longer than you think. The first time we had been working in Huntington's disease for a long time. And I remember the first time we did fetal transplant in rat in Huntington's was in '83. And the first time we did the patient at South Florida, one of the first patients was, I think, '98. So 15 years had gone by or so from the first animal studies to the clinic. So it takes a while for these. And then when you try to get a product approved, you're looking at 5 to 10 years more. So it will take a while to get these products. But what you're asking also is, what other diseases can we attack? Is that what you're asking?
Yes.
And I think for neurological diseases, we're actually approaching a lot of them. In our laboratory right now, we have trials going on in ALS, that is Lou Gehrig's disease, which is a very debilitating disease in some patients. Parkinson's disease, as I said, stroke, Huntington's disease, brain injury, which is brain and spinal cord injury is, as you know, very large in this country. And once that happens, especially spinal cord injury, you know, once you have that injury, it's very debilitating. And we actually are doing studies where we're putting in these stem cells into patients that have, or not patients, I'm sorry, into rats that have had spinal cord injury, and finding interesting results.
Amazing. We'll be right back after these messages, and we'll talk about maybe Christopher Reeves and whether there really is hope there with these kinds of discoveries. Stay with us. Welcome back. We're talking with Dr. Paul Sandberg about the end of perhaps all kinds of diseases. Christopher Reeves and his spinal cord damage. I've heard lots of people say that he's really holding out hope against hope, and there's not much chance that that's going to happen. But it would seem like with the kinds of things that you're doing that we might see him walk, that something might happen there within his lifetime.
I think it's possible. You know, and I'm usually fairly conservative in some of this stuff, but I think that the results that I've seen in our work and many other people's work using stem cells to actually go into the spinal cord injured places and regrow circuits looks promising. In fact, the first human trials now using fetal tissue have been done at University of Florida in Gainesville. We really, in many ways, the two state medical schools in Florida are quite strong in this whole area of cellular replacement in neurological disease. But I think the spinal cord injury results have proven at least promising enough that we could see clinical trials of stem cells in spinal cord within a couple of years.
Right. Has Michael J. Fox's presence with his Parkinson's and the appeals that he's made to Congress helped at all with the funds that are needed to move this forward? I mean, he really brought Parkinson's sort of, Janet Reno did a little bit, but all of a sudden with Michael J. Fox, everybody is aware of it and sort of devastated that it happens to somebody that young.
Yeah, I think he has, and I think he will. I mean, I know he has in small ways. He, in fact, came to our meeting in Clearwater this last April, and the society gave out the first award on behalf of my father to some well-known investigator who's doing excellent work. They're going to have an annual award, and Michael J. Fox handed it out, which was nice. But he also gave out 3 of his own awards. He raised some money to give out 3 grant awards to young, promising scientists in the country that submitted for the award in Parkinson's research. And that was the start of what he did. And that was before he announced his foundation. And then now he's announced the foundation. He's really gone gung-ho into trying to raise funds for this disease. And there's a lot of promise for this disease. There's already clinical trials by a number of companies in Parkinson's disease for cellular replacement. It could be one of the first that has a product to market. Yeah. We do know that it seems to help in patients. How it helps, I mean, as far as what doses should be used, et cetera, is what we have to work on next. But it looks promising.
Yeah. So many of us, we can't find our car keys or what have you, and we say it's Alzheimer's time. This ought to have an exciting potential for Alzheimer's patients too, yes?
It does. I think any disease that's degenerative, where we're losing brain cells, there's potential for it. And of course, the big one is Alzheimer's, as you said. Alzheimer's is the big one. And I think, if we all live long enough, we're all going to get it, it seems. And we, in fact, have just started a trial in the lab where we have injected stem cells into mice that have Alzheimer's. Actually, are genetically engineered these mice to have Alzheimer's disease. And we've injected these stem cells thinking that they might, if we injected them actually in the vein, thinking that these stem cells will enter the brain and go globally around the brain. So that's the thing about Alzheimer's. It's not one area that's damaged like a stroke or Parkinson's disease. It's a wide area in the brain. And so what we're hoping for is, can we get migration of these new cells within the brain?
You talked before too about stem cells from frozen embryos and the concerns that raises, but they're also harvested from bone marrow as well. I mean, that your access to them is not just one route. Right.
We, in fact, our group and 3 other groups around the country have, in fact, started to look at finding brain cells in bone marrow. There's a number of stem cells for a wide number of organs, but we actually know that there's neural cells in bone marrow, and that's a place that we're trying to look at now. In fact, we're going to be reporting this next month that we've actually found them and might be able to use them.
We just have another minute or so, but is there a way somehow, Dr. Sandberg, that the public can help in all of this effort? If it's this exciting, is there anything we can do to move it along?
Well, I think the important thing is to, contact the government, of course, if you find it of interest, because they're the ones that need to fund this at the level of the National Institutes of Health. Contact companies, I guess. There's a number of companies out there working in this, and they don't have a product, most of these companies. They're biotech companies without products. They're living on money that people hope will become something someday. But they need to just get involved. I think there's a couple websites that we have on there. One's a company one, one's an organizational one.
We do have a couple of graphics that we want to share with you. And Latent Biosciences that we talked about, you can find them on the web and look at what they're doing at latentbio.com. Or the Society of Neurotransplantation also, which is asntr.org. And if you would like to be in touch with Dr. Sandberg, you can do so by email. It's psandberg@hsc.usf.edu. edu. And I don't know how you'd find time to respond to all of those, but it's nice to offer to be able to do that for people.
Well, we'll try.
Thank you so much for being our guest and for the great work that you do. It is truly amazing. We want to thank the International Academy of Preventive Medicine for their support to this program. And until next week at the same time on Charlotte Today for SNN and Comcast, I'm Frank Cavanaugh.